The overall goal of this human preclinical model, derived from surgically resected tumor tissue, is to obtain the establishment of a primary culture of patient-derived soft tissue sarcoma.
This method can help answer two key questions about the pathophysiology of soft tissue sarcoma and represents an important preclinical tool for predicting a response to therapy.
The main advantage of this technique is that it is a fully human preclinical model.
The implications of this technique extend toward therapy of soft tissue sarcoma because this preclinical model can be tested with conventional and innovative drugs to investigate their activity and mechanism of actions.
Although this method can provide insight into soft tissue sarcoma, it can also be applied to the study of cancer cells-soft tissue cells interaction.
Individuals new to this method will struggle because of the heterogeneity of the tumor tissues and in isolating tumor cells.
Visual demonstration of this method is critical, as the initial soft tissue sarcoma cell selection and seeding steps require some manual experience before proficiency can be achieved.
Tumor specimens should be received in a 100 milliliter sterile urine container with 50 milliliters of DMEM low glucose medium sealed with a paraffin film. The samples may be kept at 4 degrees Celsius for a maximum of 3 hours after tumor resection. Sterilize the tissue culture hood with 70% ethanol and sterilize steel inox tweezers with 70% ethanol. Remove the paraffin film from the urine container and discard, clearing the exterior of the